Terrific. So hi, everyone. My name is Yuri Goltsev, and I'm the CEO of Cellformatica. Cellformatica is an AI copilot company which will help you to use AI in your daily, or maybe not daily, discovery process. And I assume most of you are coming either from academic institutions or from pharma. Cellformatica was founded by me, Nicholas Samusik, and Andrew Reck, who are all computational immunologists. Me and Nikolai, we founded a couple of companies, among them McCoy Biosciences. It's a spatial company. And Andrew is a pathologist. He's a doctor, and he's actually the person behind the inventing the whole Cellformatica pipeline. Cellformatica is able to answer biologic questions, and it is able to do so like a scientist does. And, you know, when answering it, builds hypotheses and provides precise, as opposed to other LLMs, references, and it is very useful. It starts from a question, and on this slide you can see an example of a question that was used in a breast cancer study. Usually in the question we describe, there are two parts. There's context and the question itself. In the context, which is optional, we can describe a little bit of what you are actually studying. It will help the system understand your needs. And then the question will literally formulate the question. So you can see here in the context, we described that we study, you know, breast cancer, that such and such factors could be involved. And the question is, what is involved in this gene in this process? So it looks like a placeholder, this gene, because what Cellformatica does, it takes this question and it throws at a list of genes, or a list of entities for that matter. And then it is almost like a multiple choice exam. It takes every entity in the list, and again, it could be a list, it could be genes, it could be drugs, it could be organs, diseases, whatever you want, and sees to which extent this could be an answer to your question. And in doing so, it provides interpretations. It explains how could it be an answer, raises hypotheses, and even builds validation experiments. So the Cellformatica output will contain, in the most regular implementation, it will contain a table, an Excel table with 6 columns. It will have an answer. It's a detailed text where Cellformatica builds new hypothesis and justifies the involvement of a particular entity in the question. There will be an effect score to which extent it believes the entity is involved in the question. It will have a confidence score to which extent it is supported by the literature. And it have this is a phenomenal thing, a novelty score, to which extent this is unpublished, but yet highly plausible because it was maybe working the same way in other conditions and things like that. And then it will have novelty rationale, text is justifying the novelty score, and validation experiments. This is just being currently released, and in the mainstream, Cellformatica is still not there. This is an example of Cellformatica output. You will see in the bottom here is actually the table, the 6 columns that you described, and on the top is the big text of the effect score rationale. You will see it gives a little bit of information about a protein that it found to be involved in this case in breast cancer, and at least 4 hypotheses and conclusion. And also on the right, you will see an example of the hypothesis that is able to build. I think on the next slide, I have the validation experiments. So how can you use Cellformatica? You can use it to denoise your lists and prioritize them. In other words, if your particular target is hidden within the list of candidates you did a CRISPR screen, majority of the hits are noise. So in the middle, you have your golden nugget. It can actually pull out this golden nugget. Cellformatica is trained on all ever published literature. So it can rationalize and think like a scientist and make hypotheses, but based on the available literature. So basically it will denoise your list and pull out something that according to literature, and I don't mean the direct coincidence of your entity or gene with a particular term or effect, it may be happening in a different cancer or in a different disease. So it will be able to identify by just as you read your papers and build hypothesis and provide it for you and re-rank it. Another way to use Cellformatica, and this is something very exciting, to discover if missed promising drug targets. So for example, we use Open Targets a lot. Open Targets is a database where all the genes are ranked with respect to how they could be used for treating a particular disease. In this case, you can see the ranking of the genes based on many clinical variables with respect to how are they involved in breast cancer. And they have their own literature scores. It's called, one of the scores is called EuroPMC. Basically, this is a score identifying the involvement of the gene based on, how you can find the name of the gene in the literature with breast cancer. So, and interestingly enough, when we do this analysis by Cellformatica, when we compare the open target scores with Cellformatica scores, generally we have a correlation because, of course, the genes that are mentioned in the literature, they are correlating with what we believe is involved, what Cellformatica believes is involved in disease. But also, just by using the rationalization techniques, and we trained it in it, it is able to pull out the genes that have 0 scores in open targets and justify them. And as a matter of fact, this was the gene that I showed you in the output. This was some kind of a glycosylation involved gene. This was the gene MEN1A1, which it thinks that it is involved, it might be involved in glycosylation of the drug targets or other proteins involved in drug resistance mechanism, and provides certain references for it. So literally, the path to discovery of therapeutically meaningful compounds could be as simple as a single search. The way we use it additionally is interpreting spatial data. For example, we used it to compare the microenvironments invaded by T cells, CAR T cells versus regular T cells, trying to understand what are the differences. And this network is actually built by Cellformatica. It is able to build causal regulatory networks linking genes to effects they cause, and in turn to the other genes that are caused by these effects, et cetera, et cetera. So these networks are much more readable than the ones that you get from SpringDB. I was using Cellformatica to dissect the homotypic clustering of immune cells, of certain types of immune cells. I see in COVID lungs and not in influenza. Here's this kind of clusters of this specific, cell types that distinguish the microarchitecture of COVID lungs. And what I did, I formulated a question where I described the spatial phenomena in the context, and then asked which gene, and I had a differential expression list between, but it was a bulk list between the COVID and influenza could be involved in this phenomenon. It was able to pull out CCL18 as a target, rationalize it, and it turns out that CCL18 shown by other group is involved in formation of the structures in COVID lungs. So this kind of validates its ability to de novo discover. We also can build detailed report for a given question that you have, and it will be like a 60 pages report with causative networks and diagrams and explanation of every step. That is basically what it does. So